So our next speaker is Rafael Moral from the University of Maynooth. So I'll just leave the floor to you, Rafael. We're very happy to have you here for this webinar.
Thank you, Sylvia. Can everybody see my slides? Yeah.
Yeah.
So first of all, thank you so much for inviting me to present at this event, and thanks so much for setting up an event in stats ecology. Always very interested to see what people have to show in this area. So I will talk a little bit about some Bayesian hierarchical modeling applied to wildlife monitoring. So the idea is to talk a little bit about why we model, why we try and monitor animal populations, and what can that help with in terms of conservation efforts. And why exactly are we counting these animals, and how we actually can count them, and what sort of counts do we need to try and estimate how many animals we have in a certain environment. And then I'm going to show, if I have time, two recent methods that I have developed alongside people working here at Maynooth University and in Southeast Brazil. One of them is a method that can estimate animal abundance for communities when we don't mark those animals, because one particular study is when you capture an animal, you mark the animal, release it in the wild, and do what we call a capture-recapture study. But what happens when you just sightings, you just see those animals, you don't capture, you don't mark them. So we have some models for those. And then we go up another level in terms of difficulty, let's say, is when you don't even see the animals, you only see some vestiges. You only see footprints or scats or fur, feathers, and so on. So can you still try and estimate how many animals you have if you can't even see the animals? And then if I have time, I will talk a little bit about the use of biomass as a proxy for biodiversity. I have a couple of plots. This is still work in progress. I have a few exploratory results to show that indicates to us that it might be the case that we can use this. And this would help a lot in terms of establishing some sort of protocols for wildlife monitoring that are non-invasive, long-term, and can cross different spatial scales and so on. Okay, so in terms of wildlife monitoring, do we do that? What are the reasons we should intervene in wildlife? So there's a very interesting paper that was published in Journal of Animal Ecology in the 90s by Graham Cogley. And what he basically says is there's only 4 types of human interventions in wildlife. One of them is when you try to increase a population that's in decline. So that's a type of conservation effort. The other is when you try to decrease a population that's growing out of control. So that's when you're trying to control a pest, for instance. You can also try to optimize the yield if it's a species of economic importance or food importance and so on. And when a species is not in decline, it's not growing out of proportion, you don't need to optimize yield, you just need to keep an eye on it. He actually uses that, keep an eye on it, as in terms of monitoring. Now, the issue is there's about thousands of species that need to be conserved. That are sometimes in the brink of extinction and sometimes threatened. About hundreds of species that are pests, and about dozens of species that we can try and optimize the yield for. But the other millions of species are in the none of the above category, are in the keep an eye on it category. So how can we keep an eye on millions of species without spending that much time and that much human resources and that much economic resources? And so this brings us up to the topic of the constraints that we have when you try to monitor wildlife, because sometimes you don't even know what you want to sample, what you want to measure. So these are what we call conceptual constraints. But maybe you know what you want to sample and what you want to measure, but you don't know how. So that's a technological constraint. Maybe we don't have a defined statistical model to actually try and estimate what we need to estimate from those data and so on. But sometimes you know what, you know how to do it, but you don't know who will do it, you don't know when, you don't know where, and that's a governance constraint as well. So there's a lot of different levels of constraints when you're trying to monitor wildlife, and that's why it's so difficult. And not to mention, we do have scaling effects or scale effects. So if you look at Simon Levin's inaugural talk for his MacArthur Award in ecology, where he talked about scale as the main principle surrounding ecology, he actually says that scale is the main principle surrounding all of science, because everything is dependent on scale. And in terms of monitoring populations here, we do have time scale effects and spatial scale effects. If you go from a small temporal scale, all you're going to detect are sampling processes. sampling efforts, detectability, bias, and so on. You need to measure to a certain median time scale where you can actually then identify some population level processes that would include trophic cascades, use of space, use of resources, niche overlap, habitat usability, abundance and diversity patterns. And then when you go to large time scales, that's when you identify evolutionary processes. So what are you measuring? It will depend on your temporal scale. It will also depend on your spatial scale. You can go from populations and communities to full ecosystems and landscapes to whole biomes. So again, it's not by any means an easy problem to solve. So mainly I work with counts. I like working with counts of animals or counts of vestiges, as you're going to see today. And I like to check how this abundance changes over time in space. I do like this figure from Alan Zor in his book, although I have to confess that I think his figure is wrong. So I do have a corrected version here in terms of the structural zeros and non-structural zeros. So imagine you're trying to count hippos. Well, maybe you don't see any hippos, but there's a couple of reasons why you won't see them. Maybe he's not there because the habitat is not good. You're trying to find a hippo in the Sahara Desert. You will never find a hippo there. That's a structural zero. But maybe he was there, but you just thought it was a crocodile, or you didn't see it because it was underwater, or he just took a potty break. He wasn't there but the habitat is still good for it. So that's within a count process. So this is kind of showing a zero inflated type of model, but this brings about a particular concept that we call imperfect detection. Just because you don't see the animal, it doesn't mean it isn't there. And of course, counting is very difficult. It is very hard. I have an example here with birds. So ornithologists typically go to the field and they go with a clicker and binoculars, and they look at the animals and they have to count the birds as they see those birds. So here we have a still picture. And if I ask people in the audience to count how many birds they have and I give you 20 seconds to do it, I'm pretty sure people are going to come up with different numbers. Now imagine doing that in the field when you don't really have 20 seconds to do it and you only have one shot. So counting is very difficult. So to give another example here, I'm pretty sure people know what a snow leopard is. So there you go. And sometimes snow leopards even pose for the camera. It's very easy to see them sometimes, that they even appear in camera traps like this. But I have a timer here. I'm gonna give you 20 seconds to find the snow leopard in this picture. And then I'm gonna show you where it is. I've shown this a few times. Only one person has ever found it within 20 seconds. I know where it is, but I even have to take a few seconds myself. All right, I have 10 seconds left. I'm pretty sure the snow leopard has seen you by now. And here is the snow leopard right there. So the eye, the nose, the mouth, or the ears, and the body here, hiding behind the rock, looking at the dinner. Right. So counting is indeed difficult. Just because you don't see the animal, it does not mean it's not there. So that's the problem of imperfect detection. So sometimes you don't see it because the animal is hiding or it's difficult to spot. Sometimes you may be using your tools the wrong way. And sometimes maybe your methods just have biases or inherent biases. For instance, here we have pheromone traps or color traps for insects. Maybe the insect just flies past it. Pitfall traps for small amphibians and rodents. Maybe they just aren't hungry enough to go for the bait.
Rafael?
Success. But here, oh, did I?
Rafael, you were gone for a few seconds, so
Okay. That's okay. Those weren't important seconds. It's okay. So this is a Royal Z mixture model. It's just a mixed model. I'm going to explain what it is. If you imagine you have counts of animals, those counts Y, they are interpreted as a proportion or as a proportion of the total abundance in that place. So you see Y animals given a latent abundance of N. Okay, and we assume that is a binomial. And N is the denominator of the binomial, and this is the random effect, instead of random effect being included in the probability of success. So this is the probability of detection. And then we assume that N is modeled with some count distribution, the Poisson, negative binomial, geometric, Neiman type A, whatever you want. Because we make a few assumptions here, we assume that these are counted in different sites and different time occasions. So we assume the sites are independent. Assume the populations are close, which means there's no different birth rates and death rates or migration going on. We can then write a likelihood for this model. So where we have a product of the binomials over time, we integrate out the random effect, which is the latent variable here representing the true abundances. And because we assume sites are independent, we can now multiply these together. So here we multiply because we assume populations are closed, and here we multiply because we assume sites are independent. And this model has been used since then. There's many different theoretical derivations about it and different extensions, trying to relax the closure assumption, the spatial dependence, and so on. But this model has served us pretty well in terms of animal counts. So Neve Mimna, who was working with me for the last 6 years now, she created this extension for communities now. So it's an extension of Royal's model for communities, and it's estimated in the Bayesian framework. So now we have S species, and we include this random effect A here for the abundance parameter. And this is a multivariate normal where this variance covariance matrix sigma allows us to capture correlations or covariances between species abundances within a community. And then there's, of course, different extensions for this model as well. So she proposed this as the baseline, as the base multi-species and mixture model. But then we may have, for instance, a model where we account for extra zeros. So instead of assuming a Poisson for the latent abundances, we may assume a hurdle Poisson or even a zero-inflated Poisson. So we can actually accommodate extra zeros. And we may also have temporal dependencies and relax that closure assumption. So now we have not a regressive type effect going on here for our latent abundances. And then, of course, if we can do hurdle, if we can do AR, we can do both at the same time. So now we just have a hurdle AR extension. So we have extra zeros plus that serial autocorrelation incorporated. I think one of the neat things about this model is that matrix sigma, because from this, we're able to derive marginal correlations between NS and NS prime, between two different species, or marginal covariances and marginal correlations. Interestingly enough, I really don't have an answer for this, so I'd love to hear if you have an idea. I don't really understand the full implications or the full interpretations of these correlations, because they all depend on where and what time, at what point in time you're measuring those abundances. So imagine, for instance, predators and prey, they're going to go in cycles. At some point during those cycles, their populations are growing together. Some other points, one is growing, the other is declining, and at some points, both are declining. So we may find positive and negative correlations even for predators and prey. So a negative correlation doesn't necessarily mean a negative association. It depends on when you're measuring them. So I'll just show you an example here. This is using the North American Breeding Bird Survey. We have 10 species of birds that we looked at from in the 2010s, so for 10 years. And we looked at those species of birds in the Alexander Archipelago in Alaska. So that's this region here. So we were looking at the bald eagle as a focal species, and the other species of birds as some sort of indicator species for this multi-species model. I don't know exactly the names of every single of these birds, I apologize. So this is the abundance of the bald eagle. We fitted different types of models, and the one that gave us the best in terms of information criterion was the multi-species hurdle model. We used the lat-long response surface for the abundance rate, and we used detection probability that varied per species. So here is to 2019. It stalled or halted and went down.
Rafael, you were gone again for a few seconds. So just if you could quickly go back on the previous slides. Yeah.
Okay. So this is showing estimated bald eagle abundances between 2010 and 2019 in the Alexander Archipelago. And here we're looking at a marginal abundance for the full region. And this is where we would maybe focus some conservation efforts if we need it. So in this case here, it doesn't seem like there's a need for conservation efforts in the Alexander Archipelago for the bald eagle populations, However, this last decline here between 18 and 19, we probably should investigate this further and see what happened in 20, 21, 22 to maybe make a better decision. This is kind of old now. We're 6 years ahead of time now. And this is the correlation matrix between the, and now you have the names of the birds there, between all the species. And again, it doesn't necessarily mean they have positive or negative interactions, but it might mean the conservation efforts towards one species might affect the other species as well. So that's what it means. So they are indeed interacting within that particular trophic structure. There's a package called the multi-N mix, our package. It's already on CRAN now, so you don't need to, there's the GitHub version, but you can download it directly from CRAN. All of this is implemented in Nimble. It was implemented in JAX before, but then Neve re-implemented everything in Nimble. It seems to be a bit faster to run. So that's when you can see the animals, right? But what if you can't even see the animals? What do? You just sit back and cry? Well, no, there's ways. We can actually try and find something to do here. So I'll give you an idea here. So, speaking about data scarcity, yeah, from our last talk. So imagine you have a scenario here where you have very, very scarce data. Imagine you're trying to count mallards or ducks in a certain national park, but you don't see any duck. It's a bit difficult. You would definitely see a mallard, but just bear with me. Imagine you don't see any duck. All you see are feathers or footprints. And you go in 5 different routes and you count how many times you see vestiges. So this is the data you have. I saw 6 times, twice, 3 times, none, and 7. That's all the data you have. Can you answer this question? How many mallards are there? Well, if you take a step back, you can answer the question, do we have any mallards here? Okay, so maybe transform this from an abundance study to an occupancy type study. And you can answer that. Of course, they're there. And you can answer about questions about habitat use using spatiotemporal data alongside some knowledge on mallard movement. You can still estimate abundance, and you can use some very neat methods. If you actually knew the track length, you could use what we call line intercept sampling. You also need to be very rich because you need a helicopter to do that. But imagine, for instance, that this is all the data you have. And imagine that you don't really have the time and money to invest in helicopters and very granular spatiotemporal data. What can you do then? So this is what we worked on over the last few years. And so this is about to be published in a book by the end of the year. So this is what we call the triple Poisson model. It can be a triple neck bin model. We can change from Poisson to any other distribution, but the original formulation is the triple Poisson model. We're going to make a lot of assumptions because when we lack data, we have an abundance of assumptions. And you're going to hear me say assumption here a lot of times. So first of all, we assume that animals move randomly. We assume the area or this homogeneously used. So these are 2 very strong assumptions. We assume they move around in G groups of size nk. We assume that the number of groups in different areas follows a Poisson distribution with a certain rate, lambda g. So the total abundance now is when we add all of those n's for each group. Yeah, add the number of animals for each group, and we have this value t. If we now assume again that the number of animals in each group, so this number of groups, the number of animals in each group now follows, each one follows a Poisson with the same mean lambda n. We can use some very simple results in mathematical statistics to show that the conditional distribution, total number of animals given number of groups, is also Poisson, where we have g times lambda n. Okay, but we don't observe t. We don't observe animals anymore. We observe vestiges. So now imagine that v, index t, is the number of vestiges in the environment at time t. So now let's assume again that these vestiges are produced at a constant individual daily rate that I'm calling beta. And sorry, I think you're not actually able.
Yeah, so you were just starting to introduce the the
Right, I think I'm back.
concept of messages. Yeah.
I think I'm back. Yeah, sorry.
Yeah.
This is Ed here home. It keeps crashing on me. What was the last thing I said?
Oh, you were just starting on the slides, introducing messages.
Okay, so let VT be the number of vestiges in the environment at time t. We're going to assume they're produced in a constant individual daily rate, beta, and they disappear over time. So I'm assuming here that this is the number of vestiges that are produced today. Let's think of daily data. Today, plus whatever was in the environment yesterday and the day before, and they decay exponentially, for instance. But we don't really care about this decay rate and how it goes. We're interested in a limiting distribution for the number of vestiges. So if you assume that after some period of time, that production is constant, we can simplify it and just write number of vestiges as Poisson alpha t, where alpha is the limiting rate of vestige production that we call the individual vestige surplus. So that's not too bad. So alpha for an exponential decay, it can be written very simply, like e to the delta beta over e to the delta minus 1. But we just call it alpha. We don't really care about its format. Then we have that triple hierarchy. If we know, for instance, the coverage of our sampling methodology. So we say number of vestiges given total number of animals is Poisson. Individual vestige surplus multiplied by total number of animals that we don't know, multiplied by coverage that we assume is known. That total number of animals given number of groups is also Poisson, and the number of groups is also Poisson, forming this triple Poisson hierarchy. Now, again, we need to include some informative priors here if we can, because we do have scarce data. But the good news is that for many animals, there are studies, there is empirical evidence already to suggest how many potential groups of animals we have in a certain region, and how many animals we have per group. So it's actually not difficult to find good informative priors for lambda n and lambda g. The alpha parameter, however, is difficult to inform because this is the individual vestige surplus rate. It's a bit abstract. It's not really a production rate. But anyway, we did a few experiments, and this is just a simple one. I'm going to skip through the simulations, but I'll just give you the main details. I do have 5 minutes left. Is it, Sylvia?
So 5 to 6 minutes, up to you.
Okay, so that's okay. So here's what happens when we fix different values for that individual vestige surplus rate. So here we have a true alpha of 35 and the true number of animals of 86, which is the dashed line here. If we inform the model the true value for alpha, we get really close to the true abundance for our estimate, and our precision is really good as well. However, if we overshoot alpha, if we inform alpha to be greater than it actually is, we still have a very precise estimate, but it's very downwardly biased. The other way around, if we undershoot alpha, it's going to be upwardly biased, but still very precise. If we are unsure about alpha, the good news is, and we did a lot of simulation studies about this and it is reproduced, if you give an uninformative prior for alpha and estimate everything together, you still get a very good accuracy at the expense of precision. So it's not too bad. We did a bunch of simulation studies. We were comparing with the gold standard, which is distance sampling, that can do that as well. And we actually did a nice thing. We simulated from the distance sampling model. So we were giving distance sampling the upper hand to compare with us, with our methodology. Distance sampling, we need to estimate a production rate of vestiges and a decay rate. If you have mild deviations from the true values, you start having a lot of bias in your abundance. For the triple Poisson model, the relative bias is not too bad, even when you have informative or uninformative priors for lambda n and lambda g. So that's good news. So we compared with the frequentist distance sampling and the Bayesian distance sampling as well, because it's not fair to compare a Bayesian method to a frequentist one. So we kind of did both. So we have a few plots here. This is the true abundances and is reproduced by the distance sampling that has the correct production rate and decay. But as you have wrong production rate and decay, you go down in your distance sampling in terms of your, well, this is downwardly biased. And for the Bayesian ones, you're upwardly biased for some reason. I'm not even showing the other ones because it goes up really, really high and we can see in the plot. And this is the triple Poisson model there. Not too bad. Right. So, and this is some simulation studies. I'm just going to go really, really quickly here. If you know alpha, you have more precise estimates. If you don't know alpha, you have a little bit of bias, but not too bad. You just have more imprecise estimates. Now, to finalize, so here I actually asked ChatGPT to make this figure, so that's why they look weird, but I just thought that they were interesting to show. So I said, can you give me a picture of a forest only based on latitude and longitude without the third dimension? And then that's what it did. And then I said, can you now give me the same one, but highlighting that trees have different heights of canopy heights, and we have a third dimension there. And then it drew these weird trees here that remind me of the movie Avatar. But anyway, I just wanted to show that so far, typically do, we use two dimensions when we're estimating abundances or biodiversity. But what if we want to use the, sorry.
1 minute.
Oh, good, that's enough, yeah.
Just one minute.
Yeah. What if we can actually use the third one? Because animals hide in canopies, that this represents resources and so on. So we could measure biomass and try and use that as a proxy for biodiversity. This has done in 1987 with kangaroos in Australia. So you see here, this is pasture biomass and rate of increase of populations. There is a relationship there. Of course, removing a few outliers, as sometimes it's done. This is one from southeastern Brazil that my collaborator Luciano collected. This is amphibians, so that's a biomass buffer and abundance. There seems to be some sort of relationship, and this is with small mammals. Although a bit imprecise, there seems to be some sort of relationship. So what if we can use that as a proxy? We can then use drone imagery, satellite imagery, use leaf indices such as NDVI, for instance, estimate biomass, use biomass to estimate abundances of animals or diversity of animals in different places. So reducing even more cost and making it scalable. Again, there's a bunch of assumptions we need to use, but this is a matter of being pragmatic versus extremely accurate or precise. So that's it. I'll finish with a picture of a pine marten. Thank you very much.
Thank you very much, Rafael. Very interesting talk. So there's time for a couple of questions. Is there any from the audience? Okay, I guess I'll start. I'll pick one. So, well, one is a curiosity. So when you're talking about the abundance models and the interpretation of the covariance matrix sigma, so there, I'm guessing all of the models are not fit treating the data as longitudinal, or did I miss something? Because you could put a separate prior on the covariance matrix and then try to use that, assuming that if differentiation of the covariance depends on the point in time and how they're interacting, then you could get the estimate that you want, or try at least by putting an hyperprior there and treat as the parameter of interest that overall covariance matrix. Has it been done? Is it doable even to collect that type of data to?
Oh yes, absolutely. That's a brilliant suggestion actually, because when we do the AR type, we have one correlation matrix per time point. So that helps in that regard. But that's without actually doing the actual hyperprior. It's just because we introduced this term here. This now has implications to our correlation matrix over time. But that's a brilliant idea. We can actually put a hyperprior even when we have this type of model. We can still
you put an hyperprior on that and see does it actually capture the kind of like average interaction between the species, which might be the association you're looking for. And is there any question from the audience? Rebecca, do you have a question or? No, okay, sorry. Because okay, so Simon.
Yeah. Oh, just a very quick one. You mentioned that the alpha parameter, it's not particularly easy to kind of communicate.
Yeah.
Is it actually that essential to estimate in the first place for like communicating with stakeholders at like the final results, or is it just essentially nuisance thing?
It is completely nuisance. Yeah, but
Okay.
it's nuisance that if you get it wrong, then you really bias everything.
Okay.
Yeah, but you're right. Yeah. So we were at the start, we were thinking of it as some production rate, and then we realized that it's not really a production rate. It's kind of a function of the production rate and the decay at the same time. Yeah.
Okay. Yeah. Interesting.
And there's a question from John.
Hey, John.
Oh, hi, Raphael. Thanks for the talk. Just a very quick question. You were talking about the difficulties of counting.
Yeah.
has any thought gone into the fact that if you've got counts, when you have small numbers of counts, they may actually be fairly accurate realization of the counts that's there. But as those counts increase, the quality of that data gets worse.
Yeah, that is true. Would you be thinking of some sort of weight function for
I
that?
wasn't thinking of anything. I was just wondering whether anybody thought of such things.
I don't know. I don't know, actually. That's a good point.
Which, because, you know, if I see a 0, a 1, or a 2, yeah, fine. But if I see 10, 20, oh, I start getting more
Yeah, it's actually,
suspicious.
as you go further up, it's more like interval censored than anything.
Yeah, that's sort of what you guess, with probably some digit preference going on as well.
Absolutely. Yeah. The 20s and 25s and yeah,
Well, or clever people that avoid the 20s and 25s.
true. Thanks, John.
Yeah. So I think that, well, I'll just make a further remark. Yeah, my second question was on something similar, and then now the assumption on the VT, on the, how did you call it? The occupation, when you have the occupation target, how is that realistic, depending on the variability of the, because the VT, you assume them to be constant in time, but that might be dependent on the variability.
Oh, the
of
vestiges.
data. Yeah, sorry, the vestiges. And then
Yeah.
that might be something that you can play with depending on the magnitude of the counts you're working
Yeah, so
on.
the
maybe.
reason we did that, I mean, we don't have data on vestiges over time. We just
Mhm.
have a one snapshot, the completely cross-section.
Mhm.
But we were trying to do, because distance sampling is heavily reliant on the beta and the delta here. And we were trying to do something that's not reliant on those because we don't have any means to.
Okay, fair.
estimate that. Yeah.
Okay, so thank you again very much. Very interesting talk. It was nice to have you with us. And then we'll move to the last talk for this session.